Hi, my name is Valeria, and my article is about the analysis of the antioxidant and antiviral effects of olive RPL, leaf extracts, and the pure compound used in a cancer cell model. Okay, in introduction, I will talk about cancer. Cancer is a disease that deals with uncontrolled cell division. It's important to know that cancer is the second leading cause of death in the world. I will also talk about the RPL. It's a medicinal plant that comes from the Mediterranean. Its fruit contains oleuropein, is a psychoidiol, a type of bitter phenolic. This component has been found to be a potent antioxidant. Okay, the general objective is the present study aims to assess the antioxidant and antiviral effectiveness of leaf extracts obtained from Olearpl bor sativa and Olearpl bor silvestri. In the methods, they use PCR. It is a variant of the polymerase chain reaction used to amplify the product of the DNA. It's a method that detects presence of pathogen specific in a matter and a genetic material such as viruses. First, we have the viral DNA extraction and real-time PCR. DNA was extracted using phenol chloroform solution. DNA was determined by fluorimeter analysis. A fluorimeter allows the analysis of chemical substance with molecules susceptible to fluorescence. In the second one, we have RNA extraction and real-time PCR. RNA was extracted with trisol. What is trisol? It's designed for the isolation of RNA, DNA, and proteins from a variety of biological samples. The complementary DNA is extracted from the RNA in a reaction carried out by the enzyme reverse transcriptase. We have some proteins that have been evaluated and are ICP0, UL42, and US11. They are expressed in herpes simplex virus type 1. Each real-time PCR experiment includes a reverse transcriptase negative control. The next one is about the determination of catalase activity, and it's very important because it is an enzyme with antioxidant enzymatic capacity. It is evaluated by the amount of hydrogen peroxide converted into dihydrogen and oxygen. 1 minute what its reaction rate. Lastly, we have protein extraction and immunoblot analysis. Western blotting helps to identify the relationship between an antibody and an antigen, and requires the use of electrophoresis to separate proteins and transfer them to the surface of a membrane. This study allowed us to evaluate the accumulation of viral proteins. Bueno, aquí tenemos los resultados. Es un Western blot donde aparecen las tres proteínas del virus del herpes simplex tipo 1, que es la ICPO, la UL42, la US11 y la KPDH. Es muy importante porque es como un gen de mantenimiento en donde siempre debe estar positivo, o sea, expresado para que este procedimiento tenga veracidad. Tenemos MOC, que son unas células supuestamente infectadas. Tenemos aquí el virus del herpes simplex tipo 1. Tenemos el medicamento aciclovir, que es una clase de medicamentos antivirales que ayuda a disminuir la propagación de este virus. Tenemos la oleuropeína, que es el componente importante de la planta medicinal que estamos estudiando en varias concentraciones, y podemos ver cómo en estas concentraciones no se van a ver las expresiones de estas proteínas del virus del herpes simplex, sobre todo en la casilla de la mayor concentración de la oleuropeína. Vamos a ver otro Western blot. Aquí también vamos a utilizar nuestro cultivo celular de células HeLa, concentraciones de oleuropeína en diferentes concentraciones, el virus del herpes tipo 1, tenemos la KPDH, que también sigue siendo nuestro gen de mantenimiento en el proceso. Tenemos unas proteínas que son muy importantes, que es la fosfo-PKR, la fosfo-CFOS y la fosfo-CJON. Entonces, estas proteínas son muy importantes porque al verse aumentadas significa que se está dando la replicación viral de virus en nuestro cuerpo. Entonces, si vemos en estas diferentes concentraciones, vamos a ver una leve disminución de la fosfo-PKR, que significa que al haber esta disminución de la fosfo-PKR, también van a haber menos concentración de estas proteínas, por lo que la replicación viral nos está dando al haber utilizado la oloropeína en diferentes concentraciones. Sin embargo, hay unas concentraciones un poco más detectables que otras. Por ejemplo, en la concentración de 300 vamos a ver muy poca como aparición de esta fosfo-PKR y las otras proteínas que desencadenan de esta misma. In the discussion, first we have three others, and they deduce that furthermore free radicals can cause DNA mutations and interfere with the signal transduction mechanism that regulates the replication, differentiation, and that are associated with various types of cancer development. Both studies are related because the development of cancer can be marked by the presence of free radicals and their ability to mutate DNA. Second, we have Omar, an author who studied oleuropein and concluded that oleuropein is known as an antioxidant, anti-inflammatory, and antiviral compound. The article and what the authors say are connected, as the antioxidant capacity of this compound was reflected in the study with cancer cells. Finally, John Ankem and Damoradan studied and concluded that modulation of oxidative stress by dietary agents could prevent or reduce the incidence of cancer, which is true because in the study the main risk factor for cancer is the presence of free radicals. In the conclusions we have, molecular biology is essential for the accurate diagnosis and identification of genetic variations, allowing the development of new therapies and treatments to improve the quality of life of patients. Second one, we have the research techniques proposed by molecular biology help to detect the function of the olea RPL and how it was used for the control of cancer.